In this video, we're going to start to look at some forms of active transport. So let's just go over the basics of active transport to begin with. So things that have to be actively transported are molecules that are transported against a concentration gradient. That is, they're going from low concentration to high concentration. So therefore, this is an endergonic process. It takes energy, and that energy usually comes from ATP. And you have a lot of different families of ATPases. You have P-type, which the actual transporter will be phosphorylated, and these usually transport cations such as potassium, sodium, and calcium. You have F-type, which transports protons, and these are going to be located in your mitochondria and bacterial membranes. These are used to synthesize ATP. V-types, which is an F-type but in vacuoles and acidic vesicles. A-type is transporting anions across the membranes. And ABC transporters. So ABC means ATP binding cassette, which means it just has this motif that binds ATP. This can transport a lot of different things. So there are a lot of different ABC transporters out there. So we're going to look at one type of ATPase, the sodium-potassium pump. This is our best studied ATPase. So this is found in the plasma membrane of your higher eukaryotes. That means we have them. You pump sodium out of the cell and you pump potassium into the cell. So 3 sodiums in, 2 potassiums out. You're going against concentration gradients for both situations. So you need ATP, you need water to hydrolyze your ATP. So you get your 3 sodium out, 2 potassiums in, ADP, and an inorganic phosphate. And this is what actually keeps our membrane potential, because 3 sodiums go out, 2 potassium goes in. Therefore, you have an overall net change of +1 to the outside or -1 on the inside. So the inside of your cell is negative thanks to this sodium-potassium pump. This will also help control water via osmotic pressure, that is the amount of ions in your cell compared to the outside of the cell. And if you didn't have this pump, all your cells will start to burst because of water pressure. Now, up to 70% of ATP in nerve cells is used for this pump. So most of your nerve cell energy is going to run this pump. And this is made out of 2 different subunits. You have a 1000 residue alpha subunit that binds your ATP and ions, and you have a 300 residue beta subunits. And this enzyme has 2 different conformational states, which we call E1 and E2. These 2 states have different catalytic activities and different ligand specificities. So we're going to look at how this enzyme works. So first we're going to start in the E1 complex that's bound to ATP. And this is the inside of the cell. So we're starting on the inside, and although there's arrows, this pump doesn't actually move. This pump is in your membrane. So E1 with ATP binds the sodium inside the cell, and you get a E1 ATP3 sodium complex. With the help of magnesium, your ATP goes to ADP, and your phosphate makes an aspartyl phosphate residue. So aspartic acid basically gets a phosphate attached to it. This is an intermediate state in very high energy. So we want to get out of this intermediate state. And the way we do it is that we pump 3 sodiums out. So we're in a lower energy E2 phosphate state. So we have a conformational change which releases the sodiums to the outside of the cell. When we're in this E2 phosphate form, potassium can bind to E2 phosphate. So you get this E2P2 potassium. Water will come and hydrolyze our phosphate from our aspartate residue, and we are left with E2K. This is another intermediate state, high energy, and so what will happen is that in the inside of your cell, ATP will bind potassium will be released inside, and we're back to where we started in the E1 ATP. Again, this enzyme isn't actually moving. All you're having is a conformational change to this enzyme. You're either in the E1 state or the E2 state. Now, we have another pump that's very similar. That's the calcium ATPase. So if you remember way back when we talked about muscle contractions, we said that on actin, your thin filament, you have troponin and tropomyosin. This blocks your myosin head groups from binding your thin filament, and the only way to release troponin and tropomyosin is calcium. Well, here is the here's the protein that will pump calcium to bind to your troponin and tropomyosin. So, cytosolic calcium will trigger your muscle contractions, will also release neurotransmitters and glycogen breakdown. So, calcium is very important. Inside the cytosol, you only have 0.1 micromolar, while outside the cell, you have a lot. 1500 micromolar. And so you want to maintain that balance. And the way you do that is this potassium pump. And it's the same idea. On the inside, you're in your E1 ATP phase. To calcium bind, you have a phosphate that is attached to your pump to make a high energy intermediate. You go to a lower energy E2 form, calcium leaves. You actually here bind a couple hydrogens. The water comes and removes the phosphates. You go back to a low energy E1 ATP phase and you release hydrogens inside the cell. So same basic idea as the sodium potassium pump, but here we are transporting calcium in hydrogen. That's it for this video. I will see you in the next one.